Today we're starting part 2 of our seed-bearing vascular plants. This is going to be chapter 17 of our book for Biology 102. First, we're starting off with Agnes Arber. She was an English botanist known as the Lady of Botany. She was born in 1879 and died in 1960. So in 1897, she graduated from North London Collegiate School and began attending the University College of London, where she obtained her science degree in 1899. Additional degrees included natural science degrees from Newnham College, Cambridge in 1902, and a doctorate at the University College London in 1905. She's going to work as a research assistant for plant morphologist, and she's going to work with Ethel Sargent, who's going to spend her summer holidays with and collaborate on her first major publication, which is going to come about in 1903. She's also going to teach at the University College London from 1903 to 1909, and she's going to perform research on monocot plants at Newnham College, Belfort Laboratory. That's going to be during 1912 to 1927. She's going to have her own private residential laboratory that she's going to create from 1927 to 1942. So in her nearly 70-year career, she's going to culminate over 200 publications. This is going to include 8 books, with her most widely known book entitled Herbals, The Origin of Evolution, 1912 is when it was published. Recognition for her botanical work is going to include becoming the first female botanist that's elected to the Royal Society of London in 1946, and a receipt of the Linnean Society Gold Medal in 1948. This is going to make her the first woman to receive this honor. So going into our plant tissues. Plant tissues are going to be specialized in functioning and in growth, transport, storage, support, and protection. There's going to be 4 main types of plant tissues that we're going to cover. We're going to go over the meristemic, the dermal, ground, and vascular. First off, we're going to start with the meristemic tissues. The meristemic tissues were discovered by Swiss botanist Carl Wilhelm von Nageli, and that's going to be in 1840. He's going to name the tissues in 1858. He's going to be comprised of unspecialized embryonic cells that differentiate into other types of plant tissues. And so this is going to be found within the meristem of stems and roots of seed-bearing plants. There's going to be 3 different types of meristems, and we looked at these, or we will look at these, in our different labs that we do that go along with this. There's going to be intercalary meristems. These are going to be found in your monocots, things such as grasses and corns. These are going to be also found in stems near the base of their leaves. So when you're looking at these in the lab, we will have some slides where you can view these. They promote primary growth as far as length. So this is going to lengthen the plant. You have apical meristem, which is going to be found in all plants. They're found on the tips of roots and stems. This is going to promote primary growth as far as length. You also have what's called lateral meristems. Now, lateral means sides. These are going to be found in conifers and woody eudicots. These are going to help them to grow wider. So it's going to be found in the roots and stems, and it's going to be called like secondary growth. So it shows how it's going to grow wider, and then you're going to have the other, as far as the apical meristem, growing taller. So no, lateral versus apical meristem. Next, we're going to go into our dermal tissues. Now, the dermis is going to be your outer covering, your skin. This is going to be like the dermis on your body. So dermal tissue for a plant is going to cover the plant's organs and provide protection for the same structures that are inside of that plant, kind of like the structures that are inside of you. It's going to consist of a single layer of tightly packed cells. The first is going to be the epidermis, which is going to be the outermost covering. That's going to secrete the cuticle. That helps to provide protection against water loss, UV radiation, and attacks from pathogens or pests that might go after the stems and the leaves. Specialized outgrowths of the epidermis could include root hairs. These are going to provide increased surface area for absorption by roots. This is going to be in the ground, in the soil. We have trichomes. These are going to be leaf hairs that are going to help to detect wind and protect against insects. And you have guard cells. We've talked about guard cells quite a few times in Biology 101 and in Biology 102. These are going to open and close the stomata, and that's going to help with photosynthesis. So these guard cells, you're going to have one on each side, so you have two per opening. They're going to open up during times of normal, as long as it's not hot or windy, extreme temperatures, they're going to be able to open up, and it's going to allow for gas exchange. Also, it does lose some water vapor, so that's one reason that they stay closed if it's super hot or if it's very windy outside. They're going to stay closed during those conditions to prevent that plant from becoming dried out or to prevent it from losing too much water. During those times, you will have a buildup of oxygen because it can't be released through these openings or holes, which are called the stomata, and you'll have less carbon dioxide. So the carbon dioxide will then come in, usually at night when they open back up, and the oxygen can come out. You also have what's going to be referred to, as the periderm. That periderm is going to be another form of dermal tissue. It's composed of dead, Siberian-covered cork cells that are going to replace the epidermis of those woody plants. This is going to be a very important component of bark, and it's going to help to provide protection to mature shoots and roots. So the word peri means around, and if you notice in this picture looking below, with that periderm, it's going to actually go around that outer edge. So our third type of tissue is going to be our ground tissue. Ground tissue is going to compose most of the interior portion of the plant structures, and we're going to talk about three different types of cells that are going to make up this ground tissue. We have the parenchyma cells, we have the collenchyma cells, and the sclerenchyma cells. Now with the parenchyma cells, this is going to be a term that was first introduced by an English botanist, Nehemiah Grew, in 1672. These are going to be large living cells that are surrounded by a thin, flexible cell wall. These are going to be responsible for tissue regeneration and repair. They also perform specialized functions such as photosynthesis, storage, and hormone production. So these are the ones where you're going to find your chloroplast performing the photosynthesis. They also can take that glucose and store the glucose. They often store glucose in a plant form of storage, and that's going to be starch. The hormones that they're going to produce, we're going to discuss in a little bit. Moving on to the collenchymal cells. This is going to be a term that's introduced in 1837 by a German botanist, Johann Link. These are going to be living cells that have thick, uneven cell walls. They're going to provide strength and support to young herbaceous plants. Now with the sclerenchyma cells, which is our third type, these are actually dead cells. So you do need to notice that this type is a dead cell. It's going to be thick and lignified. And so with that, it means it's going to have those tough cell walls that are going around these dead cells. This toughness, it's going to often add strength and support to older plant structures and to the vascular tissues. There's two types of these sclerenchyma cells. You have scleriates. These are going to be the polyhedral. They're going to be irregularly shaped cells. Their role at this time is unknown, so we're not sure. You're also going to have fibers. These are going to be long, narrow cells that maintain the structural integrity of dermal and vascular tissues. So in this picture, you can see a visual representation of each of these cells. So with the parenchyma cells, you're going to see that these are going to be your cells that have the flexible cell walls. Inside each one of those little green pieces is going to be a chloroplast. So they are actively performing photosynthesis. In the middle, you have your collenchyma cells. These are going to be living cells that have those thick cell walls, and notice how uneven they are. They're not going to be the same like the ones that you see over to the left-hand side. Lastly, on the right-hand side, you're going to see the sclerenchyma cells. These are going to be your dead cells. Notice how thick those cell walls are compared to the other two pictures to the left. And these are going to be very thick because they have that lignified cell wall inside of it. So that's going to give it a lot of tough structure, and that tough structure is going to help with support. With these different types of tissues, we're going to go into our last type, or our fourth type, and that is going to be our vascular tissue. This is going to be used for conducting water and fluid throughout the cells as well as nutrients. So these conducting vessels are going to be called xylem and phloem. Like we said, these are responsible for transporting this nutrients throughout the cell. The nutrients that the cell's making, remember, is going to be glucose. And those are made within the chloroplast, usually found in the leaves. First, we're going to go over the different scientists that help to discover and contribute to the finding of this vascular tissue. Marcello Malpighi in 1675 to 1686 is going to describe anatomical structures of xylem. Now, xylem is going to be the vessels that are transporting water with them and some minerals and things such as that. He discovered the movement of sugars and other solutes through the phloem. Stephen Hales in 1727 is going to describe water transport in plants. And Theodore Hartig in 1837 and 1854 is going to discover and name cellular components of phloem. Now, phloem is going to be the vessels that are going to carry the sugars or the nutrients. He proposed that phloem cells were responsible for the movement of nutrients. In 1858, Carl Wilhelm Nageli is going to introduce terms xylem and phloem. So before they were discussing these terms, but he's actually going to introduce these names and terms as far as what we're going to start to call them. Before they were just describing the structures and what was happening. Now, he's going to discover that these tubes actually exist, and with xylem, it's going to permit the flow of water and minerals. It's composed of two types of sclerenchyma cells that are reinforced with lignin. We have the tracheids, which are going to be the narrow tube-shaped cells that consist of small pits, and we have the xylem vessels. These are going to be short, wide cells that are stacked to create a continuous tube. Now going into phloem. Phloem is going to consist of slender, elongated parenchymal cells known as sieve elements. Sieve elements are going to possess sieve plates. That's going to help to distribute different molecules such as sugar, proteins, and nucleic acids. They're attached to the sieve element and are called companion cells. So they're believed to control cellular physiology and synthesize proteins that are going to be related to phloem transport. Now looking here, we're going to see the actual picture of the xylem, which is going to be the microscope picture that's in the circle up top, that's going to be on your left-hand side in pink. So you can see the tracheids, and you can see those parenchymal cells that go around it. So that's going to be responsible for transporting water and minerals through these vessels. So this is what's going to make up those vessels. And if you look down below, you see an artist's rendition of what this actually looks like. So it's going to have those perforation plates where the water and the minerals are going to have to flow up through it. And so it's going to flow laterally. Phloem, on the other hand, is going to be on the right-hand side. And so you can see how those cells are going to be made up. You're going to see those sieve tubes in the sieve plate. So notice how it's going to filter out. The phloem, remember, is going to be responsible for transporting sugars and other items. So if you have angiosperms, the sieve tube element is going to contain the sugar solution. So sieve tube cells are going to be surrounded by various support cells, and you can see those here. We have the transfer cell wall ingrowths coming in. You've got the plasmodesmata that's going to be on the sides, and you have those intermediate cells that are going to be around it. And so that's all going to help to bring in the sugar that you see that's going to be coming from those chloroplasts in the cells to the side. And with phloem, it's going to flow downwards towards the other portions of the plant, down towards the root. Xylem is going to flow up, so it's coming from the roots up to the tips of the leaves. So xylem flows up, phloem flows down. So precisely designated structures composed of specialized plant tissues that are going to be responsible for support, growth, and development. You're going to have three main types of plant organs here. So with these, you're going to have roots, stems, and leaves. Roots are going to perform most of the necessary functions underground by maintaining stability and absorbing and transporting nutrients from the soil. There's two types of root systems. You have the fibrous root system, which is going to be found in monocots. These are things like your corns and grasses we mentioned before. They're going to be characterized by numerous slender roots of the same similar size, and they're going to branch out. And you can see this fibrous picture down below and see how they're branching out. Taproots, however, are going to be found in gymnosperms and eudicots. These are going to consist of a large, thick primary root with lateral secondary roots. So you see that main root that's going to come down through the center, that's referred to as your taproot, and then you'll have smaller roots coming out from that main taproot. So going into the anatomy, you're going to have the epidermis on the external part. That's going to form projections or root hairs, and you're going to have a root cap that goes around the tip. So looking at that picture, you can see that that has a root cap that's going around the end. And that's kind of the apex or the hump or curved part that's going to be the deepest part inside of the soil. That is going to be a protective structure, and it's going to contain apical meristem there. Internal anatomy of these roots, if it's a monocot, it's going to have ground tissue that's divided into cortex and pith by a ring of vascular tissue. If it's a gymnosperm, it's going to compose a vascular cylinder that surrounds a central pith. If it's a eudicot, it's going to have a vascular cylinder that's going to dominate the center of that root. And so you can see all of these below with these different pictures here. So the first picture that you're going to see here, over to the side, this is going to be your monocot. You can see how it has the cortex on the outer portion. It's going to have that vascular tissue ring that you're going to see going through this area here, and then in the center you're going to have that pith. Coming over to the next one's your gymnosperm. You're going to see there that you're going to have that vascular tissue that's already circled there with the xylem and phloem. It's going to have that pith towards the center, and then you see that cortex that's going around the outer portion. So with that xylem and phloem, we do call that, remember, the vascular cylinder there. And with the last one, that's going to be your eudicot. It's going to have its vascular cylinder, which is going to go around this central portion. This one is actually shaped like a wheel. It's going to show you your phloem and your xylem that's going to be there. You can have what's called modified root structures. These are going to help with storage. You can have storage roots, which are going to be things such as carrots, beets, anything that is going to be able to store these carbohydrates below ground. And so as it's storing these carbohydrates and water and food reserves, they get stored in the form of starch. So that's how plants store carbohydrates, as starch. You could have aerial roots. These are going to be things like ivy and orchids. Those roots are going to grow above the ground. You can have what's called a prop root, like in corn plants. That's a modified aerial root that's going to originate from the stem, and it's going to help to actually anchor that plant in. You can have what's called a buttress root. Example of this would be like a tropical fig tree. That's going to be a large, wide root that's located at the base of the tree to provide additional support. And then you've got pneumatophores. These are going to be things like with the mangrove tree. It's a modified aerial root that's going to specialize in diffusion of oxygen into the roots of the plants that grow in oxygen-poor environments, such as in coastal wetlands. And here's your example of different storages. So you see that carrot there. That's going to be like a main root that's going to come in and store those carbohydrates. The aerial roots, which are going to be above ground. You've got your prop root, like what you would find in things like corns. You've got your buttress, and then you've got your pneumatophores, which you're going to find in your mangrove trees. Heading on into stems. Stems are going to be part of the shoot system. It's going to provide structural support and nutrient transport, and it's going to be arranged in a series of alternating functional units. These are going to be known as phytomers. The node is going to consist of the meristemic tissue that promotes the development of new plant structures such as leaves, stems, or flowers. There, you're also going to have an internode. That's going to be the region between the two nodes. An auxiliary bud is going to give rise to new stems or flowers. And so you can see the auxiliary bud, that's going to kind of be where that stem's coming off, looking at the picture. And then you're going to have different plant tissues of the stem that are going to arise from the apical meristem, which is going to be on the top. That's going to be protected by a terminal bud that's located actually on top of the whole structure. So, hold on just a second. Bring my pencil up. So all of this is going to be referred to as the terminal bud. Inside of it is going to be that apical meristem that you see. It's kind of protected by the little sprigs that are coming up on each side. We talked about the axial bud. That's going to be found coming off right here with the stem that's coming out. We're going to have a node here. We'll have a node here. And in between these two nodes, you're going to have the middle section, which is referred to as the internode. Now, putting all of this together, the axillary bud, the nodes, and the internode, we're going to call this a phytomer. Now we're going to go into the anatomy of a stem. So starting with the external anatomy, it is protected by the epidermis that secretes the cuticle. Now remember, the cuticle is going to be a waxy substance that's going to help to protect and keep in moisture. In gymnosperms and woody eudicots, the epidermis is going to be replaced by what's called the periderm that's produced by the cork cambium. And you can see that in the picture below. Internal anatomy of stems, you're first going to have your ground tissue. If it's a gymnosperm, the cortex is going to consist of outer layers of lignified sclerenchyma cells and an inner layer of collenchymal cells. These are going to have a centralized parenchymatous pit. So that parenchymatous pit is going to be found in the center, looking at that picture on the left-hand side. So that shows you your gymnosperm. The herbaceous monocots are going to consist of undefined regions of parenchyma cells that are going to be reinforced by layers of sclerenchyma cells. And so that's going to be the one that you see over to the right. Herbaceous eudicot ground tissue is going to be divided by a ring of vascular tissue into an innermost pith, and that's going to be made up of parenchyma cells. The outermost cortex is going to be made up of parenchyma cells and collenchyma cells. If it's a woody eudicot, which is what you're going to see to the right-hand side, the herbaceous was what's on the bottom left. The woody eudicots are going to consist of a centrally located pith, which is made up of parenchyma, and then you're going to have a thin layer of cortex, which is made up of a mixture of parenchyma and collenchyma cells. Just inside of that, you're going to have what's called the cork cambium. Internal anatomy of the stem with this vascular tissues, when you're starting with the gymnosperm, it's going to form a concentric ring that's going to divide the vascular cambium. And so you can see that in the picture on the left-hand side. Now, if it's a herbaceous monocot, that's going to form bundles with no distinct pattern. That's what you see on the right. Herbaceous eudicots are going to exist as a ring, whereas woody eudicots form concentric rings that are going to divide by vascular cambium. So differentiation of cells within vascular cambium is going to produce secondary xylem and secondary phloem. This is going to promote secondary growth and the formation of wood and bark. So remember that secondary growth means it's going to be growing wider. So that's width. Some of these modified stem structures that you're going to have are going to be referred to as stolons. These can be found in strawberry plants. These are stems that grow along the surface of the ground. You could also have rhizomes, like in ginger. These are underground stems that grow horizontally. Tubers, like in potatoes, are going to be enlarged regions along a stolon or a rhizome that's going to store nutrients. Remember, it stores its nutrients as starch. Thorns, like in the honey locust, are going to be outgrowths of the stem that are modified to be sharpened points that's there for protection. Tendrils are going to be like in the garden peas. They're elongated thread-like stems that are going to be used by the plants to climb and coil themselves around an object for support. And then fleshy objects, such as the cactus, are going to have thickened stems with tissue modified for storing large quantities of water. So you'll find these in areas that are going to be dry or very windy. Sorry. All right, so this shows you an example of each one of these. So take a second to look at them. Next, moving on to leaves. Leaves are going to represent the secondary component of a plant's shoot system. They're considered the primary photosynthetic region or photosynthetic organ. Two types of meristems are going to be found in leaves. You're going to have what's called the plate meristem and the marginal meristem. These are going to initiate leaf tissue development by coordinating cell division produced in the main leaf structures and determined at the point of which the cell proliferation switches to cell differentiation.